Nice to see so many of you here. I think we're going to have a very, very interesting evening. Welcome. This is our annual Mylar-Geary lecture. Mylar-Geary was a professor of physics here many years ago who passed away at an unfortunately young age. We dedicate one lecture a year, typically in the physical sciences, in his honor because Mylar was a physics professor. But we went a little bit off script this year, and this year's presentation it's not just about biology and medicine, it's about constructive relationships between citizens and science. And you read a lot about those these days. If you're just a common citizen, you can help NASA track meteors that might eventually hit the Earth. You can do crowd computing that requires massive numbers of people to work on data for various research projects. There are all kinds of things that citizens can do to move the world of science forward these days. And it's appropriate, this is a Mylar-Geary lecture because Mylar was very passionate about involving non-science majors, involving people in general in science and research. So we dedicate this evening to the memory of Mylar-Geary. I will introduce our second speaker later, but our first speaker, as you probably know because you read the publicity, is the grandmother, nobody can believe it, of one of our students, Zaire Thompson, and so Zaire is going to introduce his grandmother for us.
Hey guys, I'm really glad you can all make it out and support someone that really means so much to me. And it's really important because not only is she advocating something that's really important to her that she's passionate about, it's also my birthday. So, and who else wouldn't want to have their grandma here in front of all of their friends on their birthday? So. That is true, I'm 19. But it's not about me, it's about Sandra Harrigan-Thompson here that's going to be giving her lecture. So
thank you. Thank you for coming. Thank you, each and every one of you, for coming out here tonight. I can't believe it. I'm standing here in front of each and every one of you. I never graduated college. I went to college twice. I do a lot of things in twos. I never made it out, though, but here I am in front of you in front of Penn State. And because today I'm here, I chose this day to be here, April 24th, not only because it's my grandson's birthday, because that day, 18 years ago, I wanted to have one picture. Well, there was a couple of pictures I was going to show you, but no need to show you any pictures. You can read my book later. I mean, we can get that, no problem. But 18 years ago, when I first held him in my arms, and I do have that one picture, was the very moment when I realized that I was still alive, that I had been alive for 10 years, living with HIV. My story is a lot different than most people living with HIV, because I am one of those unique people. Throughout my whole entire life, it's been just anything, all the uniqueness, of course. Always different than the rest in one way or another. Sometimes good, sometimes not so good, but always different. And it was 1989 when I realized, here I am, still here. Okay, I can give you my story now briefly, but I gotta look at a couple of my notes. December 1989, I was diagnosed as being HIV positive. It was a total surprise to me, because I had no idea back in 1989 that I could be HIV positive. I had a child, and my husband and I we weren't married at the time, actually. 89, we were married. We got married in 87. But in 89, yeah, we got married in 87. I forgot about that. But anyway, I was just trying to talk about him, but God rest his soul, he passed on. He passed on 6 months after I found out that I was positive. That was a surprise to me because I didn't know he used IV drugs. I had no idea about IV drugs. We smoked a little weed, we did a little something else, But I didn't know he did anything else beyond that. And apparently he did, and he contracted the virus. What can I say? When I found out, I had to raise my daughter, who is here today also. And all I could think about was, how am I going to raise this child of mine by myself without my husband? First of all, I wasn't kid-friendly at all. I'm still not really kid-friendly, but I'm a little better now since I got kids and grandkids. But at that time, I really wasn't. I didn't know what to do, but I knew I had to do something. So after burying my husband, which was a trip, because I never told the family. No one knew that my husband died of AIDS. No one knew that. I kept it a secret. I felt at that time no one knew. I didn't know. I didn't know any answers about anything. All I knew was people were dying, people were getting sick and dying, and I probably was going to die too. And I had to take care of my child and provide for her and make sure she had whatever she needed to go on in life. It was a challenge to keep my mouth closed, not complain, and not say anything. But being that my circumstances were a little different, after my husband's funeral, I'm bouncing around. I should be looking at the notes. But wait a minute. Let me look at the notes. Yeah, because I done got Happy there with that. Oh, that's my nickname too, Happy, H-A-P-P-I. One of the nicknames. Back in the day, it used to be Joy.
Bouncing around is fine.
I'm sorry?
Bouncing around.
Okay. I was 33 years old, and my husband was 35, and I was totally baffled as to why he was sick for 3 months. 3 months he had been sick in the hospital. No one knew what was wrong with him. This was a former Golden Glove boxer. This was a big guy. He was tough, strong, powerful, dynamic. No joke. He was Taurus, another bull. They don't play. He didn't play at all. But here I am, having to deal with burying my husband. There were some other circumstances around. Y'all have to read that in the book too. But after I buried him, at the funeral, he wanted a female undertaker to, say the final words for him before he went on to his resting place. And her last words were, "Let's go on with this business called living." And that's what I chose to do. I chose to go on living, every single day, living. I paid HIV no mind. I knew I was going to die, because back then in 1989, people weren't living. I saw a lot of people die. I saw a lot of people suffer, get sick, their hair started changing, their complexion started changing, they started dying all around me. And my circles were sort of different. I had 2 jobs, I worked, I partied a lot, because now life is short. Let me just live it one day at a time. I partied, I worked 2 jobs, 3 jobs, doing whatever I possibly could. And I paid it no mind until 10 years. 10 years when I held you is when I was like, oh my God, I'm living with this disease. Still alive and living well. Never sick, never a cold, never a flu. Maybe I got a flu shot or 2, but I don't get sick. I don't get like that. I don't get colds, I don't get sick. If I get a cut, it heals immediately. Instantly. I'm not a Wolverine, but these are some of the things that people like me, HIV controllers, that we found through science, have these certain things. They're undetectable, never have to take any medications, never a viral load, none of that. So therefore, I am different in the HIV/AIDS community. I'm in 1% of the category. I am an elite controller. I said it out loud in front of people without fear. Because in the HIV/AIDS community, back in the early 90s, I started going online in a couple of different chat rooms and trying to find some sort of camaraderie, trying to find some people I could talk to and understand what was happening with me and my body. Oh, here's one. Back in 1991, early on in 1991, I got Bell's palsy, and I thought that was a stroke or a sign of some sort of deterioration, and I was tested for HIV again, and once again I was positive. Repeatedly tested over the years, over and over and over again. Still, yes, you are HIV positive, but undetectable, No viruses is seen, nothing, but you are HIV positive. Now, in the HIV/AIDS community, I found some chat rooms to go to, some people to communicate with, and some people were nice, they were welcoming, and everybody has their own situations, their own life challenges, and whatever. But what happened to me was, of course, being that I'm a little bit different, when I decided to open up and expose myself to everyone and let everyone know, yes, I am positive too, I realized I was different. I didn't want to say it so much, but then someone would say, well, how many T cells do you have? Or what's your T cell count like? And my counts have always been 1,200, 1,500. At that time, back in those days, it was like over 2,000, astronomically high numbers. People living with T cells like 20 T cells or 15 or lower or whatever, nobody has this number. So how could I be braggadocious and say, I have all these T cells? I was not the one to say that. I was not the one to brag about anything about me at all, because I realized I was different. Once again, different. I got beat up in some of the chat rooms because I was different. First of all, here's the scenario. I sort of understand now why I got beat up. Imagine this. Someone says, I have cancer. I say, oh, yeah. Wow, that's terrible, man. You got cancer? Well, me too. I got cancer too. Well, listen, like chemo really messes you up when you lose your hair, your nauseous, you're throwing up, and you're having a hard time. Isn't it chemo? Isn't that rough? And I got to say, no chemo. Never done chemo. Never had to. What? No chemo? Okay. Then you must be on that radiation thing. You must do a little radiation, get them radiations. Yeah, you say you have cancer, right? Yeah. But you don't do radiation either? No radiation. Oh, okay. So you must be one of the people in the clinical trials and stuff doing the new pills or something, right? No, I don't take no pills. Never took a pill. Never had to. I'm not doing nothing. You ain't got no cancer. You ain't got no cancer. You can't have no cancer. You ain't suffering. You ain't sick. You ain't, no. You lying. You're fake. You're this. You're that. Oh, no, but really got beat up. So let me keep my mouth closed. Why should I say anything to anybody? Because I'm going to get beat up? They keep going to the doctor, they take my blood, they say I'm HIV positive. But I have nothing to show for it. I don't have any symptoms. I'm not sick. I'm extremely healthy. I'm healthy as an ox. I'm real healthy. Trust me, I'm healthy. But it's okay. Because with that beat up, I stay quiet. I'm not saying nothing to anybody no more. Because why am I doing that? No need. I don't like abuse. I'm not into anything like that. I'm a happy person. I like to have a good time. I don't like getting beat up. So I stay quiet. Until Oprah decided to do that thing in Africa, save the African children, let's stop AIDS in Africa, let's cut it. When she went on that mission back in 2002, I said, okay, I've been around. 1989, I was diagnosed. I'm still here. It's 2002. 1999, I got a grandson. I'm still alive. It's 10 years. All right, let me just throw myself in there. Let me write to Oprah. I don't really want to go on TV. Not really. But if she wants me, I'll sit on her stage, and she ain't going to give me no car or nothing, but I'm going to go. I'm turning myself in. So I wrote her. I did write her. And I put my heart out. I cried writing too, because I'm putting myself out in public. I'm face to face now. I'm exposing myself as being HIV positive. I've been living with it for many years now, 89 to 2002, however long that is, I've been living. So I wrote to Oprah, because I said, she's the one that's going to listen to me. She got everybody else on TV. And this is hot topics, because, I'm here. It's me. I wrote to her, and I sent $100. I didn't have that $100. Trust me, I didn't have the $100. But I told the story, and I wanted them to listen, so I sent the $100 too. I got back an email, said, thanks for the $100. That's all I got. Till this day, Oprah and her producers haven't called me yet. I'm still waiting. Matter of fact, I'm not waiting for Oprah no more. I'm here with you guys now. I'm telling you my story. And right now, for you guys gonna read my book anyway. Because I'm an ordinary woman living an extraordinary life. Trust me on that. There's so much to say about that. But I want to talk about HIV and AIDS, because that's why I'm here. Y'all gonna read my book. It's gonna be on Amazon soon, not too long from now. The reason I went to the doctor every 3 months. I found a doctor, I was an old family doctor, and I had been going to him. He was not an HIV doctor. He was just a regular general practitioner. Been there for many, many years. My family was going to him. So I decided to go when I got the Bell's palsy back in 1991. And from then on, I started seeing him. This is hard for me right here, because when I realized that I'm not the sharpest tack in the box, and for 20 years I allowed this provider to see me once every 3 months and take vials and vials and vials of my blood, for what reason? Nothing. I have a little diabetical, a little diabetes, a little high blood pressure, a lot of anxiety, and a lot of something else. But no reason to take all those bottles of blood from me. I'm finding this out in recent years. My provider did not provide the kind of support I needed. My provider never gave me any kind of anything, but just kept telling me to come back. Every 3 months, you're HIV positive, you have to have your blood checked. So me, I need to have my records. I need to look at everything. I want a copy of all my stuff. And I don't know what I'm looking at, but I know there's nothing happening. I feel good. I'm still living life. I'm still out and about. I'm still doing everything that I always wanted to do. What's happening? But since I'm not the sharpest tack in the box, that didn't even kick in until around 2012, when I wound up meeting some people and going to a support group. And a speaker came in, and she was speaking, talking about HIV and AIDS, and the group of people that I was with, all of them were either had HIV or AIDS for long periods of time. They were all on medications. No one had my story. No one had my story of never taking a pill, never having a medication, never having an antiviral. No one in that group. I went to several groups like that, and I would get up and say a little something, but I could never tell my story because, oh yeah, yeah, you got HIV, huh? Well, good for you. But see, now, because of the Zephyr Foundation, that's why I got to talk about that. Because of the Zephyr Foundation, I found a niche. I found some other people that were just like me. Long-term non-progressors, people that have been HIV positive for very long periods of time, not taking any medication, not having any of the normal routines that people go through that have HIV and AIDS. The Zephyr Foundation. A woman by the name of Laureen Willenberg. You will be hearing her name again, remember that. Laureen Willenberg, the founder of the Zephyr Foundation. She's the one that had the insight and the genius to create a place for people like us. 0.1 point of the population of 38 million or more people living with HIV and AIDS, we're in 0.1% category. Just us. Known HIV Elite Controllers, such as myself, it's only a little bit over 500, as far as we know, probably a few more. There's probably thousands of us, but people that are like us don't get sick. You don't need to go to the doctor. No need to get tested. No need to, if you're not doing that, you don't know. You have no idea. We just know about, researchers really depend on the Zephyr Foundation for people like me, to find people like me that are willing to come in out of my own time, on my own time, to do whatever I possibly can do for the cause, to end HIV and AIDS, find a cure, find a vaccine. That's why I do what I do. That's why for the last few years, I've gotten in touch with the Zephyr Foundation and Laureen Willenberger, and because of her, she gave me the courage to be here in front of you today. Because of her, she went back to school. Matter of fact, she's graduating May 18th, from college with her bioethics degree. And I'm so proud of her. At my age, she's 63 also, and she's in California, and she's doing her thing. I can't imagine going back to school to do this. I can't, because I can't focus that long, as y'all can tell. But God bless her for her having the wisdom and the insight to create a place for us. My purpose right now is to do the best I can to help anybody that needs help. I'm on a mission. I think it's from God, honestly, because He's kept me here and kept me doing good, kept me well, kept me extremely healthy. Although I like being called small, I like being called subset, because I'm unique and in that category, but I have to do whatever I can do to help research and scientists. And basically, my mottos are, be good, do good, and good will come to you. And Helping others is what my life is all about. I can't imagine doing anything else. I can't imagine not doing something for other people. I can't imagine my day not making somebody smile. I can't imagine my day not making somebody happy. I can't imagine my day not giving something back for this gift that I have within me. I can't imagine it. So I smile all the time. Whether I'm happy, sad, glad, or mad, I got a smile on. Sometimes I think it's like a pseudo-barber, some kind of new condition because I'm laughing, smiling, and mad at the same time. But I keep the smile on because it reminds me this is what I'm supposed to be doing. And this is what someone sees when they look at me. They see the smile on my face. I think it holds back the wrinkles too, actually, keeping that. I'm going to wrap this up a little bit here. But my final thoughts to you, one more time, is to always be aware that somebody needs you. Somebody needs you out there. They need your support. They need your love. They need your kindness. They need your help. So be kind. Be giving. Be generous with yourselves. The more you give, the more you get back. Trust me, I'm 63. I done found out. I give more than I ever gave now. Trust me. Not for the reason of getting back, but because it's good. It's a good thing. It's a good feeling for me. It gives me inner happiness and inner peace. I encourage each and every one of you, if you've never been tested, get tested. Just get tested. You just need to know your status. And the other thing is, help stop the stigma about HIV and AIDS. Educate yourselves. Learn more. Oh, matter of fact, you're going to get an education in a few minutes. Wait till Dr. Migueles gets up and gives you all the details and the stats on what's happening now in the HIV AIDS community as far as immunology is concerned. Stigma is really horrible. And I've joined several support groups and several people. I talk with lots of people, and I hear lots of voices now. They're almost in my head. I hear people crying for help, crying for assistance, crying for compassion, crying for an understanding, crying for people are dying, still dying of HIV and AIDS, often. People are committing suicide because they're not getting help when they have these disease that they're being stigmatized for. When you go home and you tell your family, I have HIV. Depending on some of that family is going to be, oh, don't touch my cup. They're still on that ignorance from back in the days and whatnot. There's some things, but educate yourselves. You're here. You're all beautiful, wonderful people. And thank you so much. That is my part for now. And I'm going to turn it over to Dr. Migueles.
Maybe you want to tell us how you and Dr. Migueles got together?
Oh, I missed that part. Oh, wait. Well, it all comes back to the Zephyr Foundation, meeting Lorraine Wollenberger. I don't even remember when this happened. It's about 5 years ago now. 4 or 5 years ago? 5 years. She told me on to the National Institute of Health. Matter of fact, I had reached out to Harvard, Mass General. They said, oh, yeah, come on. We want you down here. I sent them my stats and my bios. They said, oh, yeah, come on. We'll give you $500 for a colonoscopy. Oh, okay. But Lorraine, I'm sorry, pardon me. Lorraine is very knowledgeable. I didn't go. Okay, fine. We'll talk later. Lorraine said their bioethics, their ethics, and all the consent and all that is not quite right. The wording, I don't know what that says, sign here. So she said, don't do it. She said, go to the National Institute of Health, get in touch with Dr. Stephen Migueles, get in touch with the NIAID team, and they will do right by you. They have all the legal wording properly. Your rights are protected. You're not going to be abused. It's all in writing. I've already done it. She's already done it. She's done so much. She's done a lot. She's been years and years. I can't even remember her story and how many years she's been there. So she knew. I followed her. And I followed her completely because she and I are 63 years old. I'm black, she's white. I'm on the East Coast, she's on the West Coast. And there's not too many similarities between us as far as our medical status. So when she referred me to NIH and I was introduced to Dr. Stephen Migueles, I found it. That was it. I have a purpose now. I got something to do. And I got somebody that's going to help me get there. And I'm so happy to say that Dr. Stephen Migueles has driven from NIH just today, just to be here with you guys. But because of me, because he can't hear. But the message is powerful that this man has for you. And I'm so excited to introduce Dr. Stephen Migueles from the National Institute of Health. Thank you.
Dr. Migueles, investigates the immune responses in HIV-infected individuals who exhibit durable, long-lasting immune-mediated control over HIV replication without antiretroviral therapy, which is a fancy way of saying people like Sandra who are controllers and who don't show any signs of the disease. He has been an associate investigator of multiple intramural NIH projects related to these unique individuals who are called Long Term Non Progressors or Elite Controllers. He has identified key features that constitute an effective anti-HIV immune response in order to inform the novel design of the novel design of efficacious HIV/AIDS vaccines. What that means is that people like Sandy provide us information on the way the immune system responds to attack by HIV that can be, we hope, very useful in developing therapies or immunotherapies, developing vaccines. And it's that promise about there, which is, I don't know if you know, but Zika virus came out and it was a big problem and we had a vaccine within a year. Developing vaccines is kind of become easy, but HIV is very, very tricky. We've been looking for a vaccine for HIV for very many years, and nothing has worked. And I imagine that's if it were for Dr. Migueles, that this would be a tremendous accomplishment for him, for the people at NIH, and for the people like Sandy. Dr. Migueles.
Thank you. That was really kind, thank you. Imagine my terror when I realized I was going to follow Sandy in a presentation. My heart rate got up a bit after that. But let me just say, as the NIH person, I'm supposed to know something, and the one thing I can tell you with 100% certainty that I know is that you are not ordinary. Nothing about you is ordinary. You're an extraordinary human being. So, thank you. So what I'm going to do, and we talked about this a lot, and I struggled a lot with how I wanted to approach this. I want to take just a few slides and talk about HIV itself, the pathogen, and I'm going to kind of work from the microbiologic level up to the host and talk about Sandy. And I'm going to be I have a few cartoons and I have a little bit of data, and I don't want it to be too science heavy, but I want to tell you the story of what we've been struggling in our lab to figure out in the last 19 years. All infectious pathogens, in order for that organism to propagate itself and propagate its genetic material, it has to be able to get into a cell, and to do that it's got to bind to a cell. And there's some molecules on the outer coat of HIV that are responsible for allowing it to bind to immune cells and get in and infect those cells. I first joined NIH a long time ago. I was looking for a lab. I met my boss. He said, I have 3 people whose cells I've been studying. They're 3 white guys. They're Irish American. Their last names begin with Mick. They've been alive for 15 years. They have normal CD4 counts. Viral load assays were just becoming standardized. We can't detect the viral load in them. I don't know what to make of them. And I remember hearing this story and thinking of a woman that I took care of that I admitted to the hospital when I was a resident at Georgetown, who I knew was very sick, had an acute retroviral syndrome. She got better in the hospital. We ruled out everything else. We diagnosed her with HIV. I followed her in my clinic, but we could never measure a viral load on her. And all the ID people, the infectious disease people whom I really respected, said, she must be a false positive. It's probably the wrong diagnosis. I said, how can it be the wrong diagnosis? We repeated the test, it was still positive. So I heard my boss saying that he had these 3 patients, and I remember thinking at this moment, this is the Holy Grail. If there's somebody who naturally, spontaneously is controlling the virus or has cured themselves of the virus, we all should be studying them, because then we can figure out how they do it. We can identify the correlate of protective immunity, and we could figure out how to vaccinate other people or to use this to treat other people, maybe even other diseases that have an immunologic basis. So we started doing that. We came together, the 2 of us, we wrote a protocol, and we started the first trial at NIH in order to, we started the first trial at NIH where we were going to specifically recruit patients like Sandy, who were very rare, and study their immune system. So let me get back to this. So I'll edit that part out to keep this timely. These are the receptors. This is GP120. So in this retrovirus, 2 RNAs encapsulated by this nucleocapsid, which is responsible for protecting it, covered by an envelope that has these knobs. And on an electron micrograph, when you look at the virus, it actually has these spokes on the outside. This is glycoprotein 120. It's a sugar protein. 120 is because of the molecular weight. This is how it gets into a cell. This is the genius thing and the frustrating thing, that this virus has co-evolved so that it can bind to CD4 positive T cells. So that's what's shown in the next slide. So here we have the CD4 molecule. This is on one subset of T cells. T cells are specialized white blood cells in the body. They get educated in the thymus to figure out what's self and what's non-self. And they spend their life percolating the body, looking for things that are foreign and try to mount an immune response against it. So this molecule is what GP120 binds to bind to a cell so it can infect the cell. The crazy thing about it is, as you can imagine what I was alluding to before, is it's basically attacking one of the very cells that is responsible for defending the host against this virus. So it's really part of the conundrum of HIV. Once that happens, there's a change in this GP120, and then it binds to a second molecule, a co-receptor called CCR5, which we'll come back to. When that happens, there is a fusion event, where now the envelope of the virus fuses with the host membrane, and the nucleocapsid gains access into the cell. So now it can propagate its genetic material in the cell. Basically, it converts the cell into a virus factory. The goal is to create a lot of daughter viruses or progeny virions that then get liberated from it. So this is the life cycle that I'm not going to go through these steps, obviously, but we've learned a lot from understanding this. And one of the benefits of learning this, following the tuberculosis paradigm, is that if we interrupt more than a few of these steps, we're more likely to bring the virus to very low levels and give people a normal lifespan, because we're cooling off the inflammation, cooling off the immune activation, keeping the virus to very low levels. Sandy doesn't need these meds, so we're going to get to why she's able to not require them. But this is basically how the virus gets in, starts to infect the cell, and then it amps itself up. When this cell bursts 24 to 36 hours later, tons of these things come flying out and they infect everything in the vicinity. Okay, so this is two more slides of intro, and then I'm going to tell you a couple specific things. This is showing basically a breach in the cervicovaginal epithelium with the virus coming in and infecting CD4s that are there. There's also some cells like dendritic cells, and dendritic are these dendrites, these arms, that can reach out and help pull the virus in, and they can actually hold it bound into little vesicles within the cell itself, and they bring it to the lymph node. The jobs of these cells is to sample what's around and bring it to the lymphoid tissue so it can start an immune response. Well, imagine, that's like bringing your worst enemy to your weaponry arsenal and showing them where all of your weapons are. So you're bringing HIV to the very cells that it likes to infect, and it bounces along and it picks the cells off, and then it amplifies itself and it replicates to very high degrees, and it disseminates through the body. This is over hours to days to weeks. This is a problem because this is occurring in an unrestricted manner. There's nothing else trying to stop it. So you have HIV interacting with the immune system, but eventually the immune system kicks in and tries to fight HIV. And that's what's shown in this panel. And this is a setup for the 2 or 3 science slides I'm going to show you. These cells that are shown here in gray, these are called macrophages or monocytes. These are professional antigen presenting cells. These cells endocytose, phagocytose, take in material, process it, chop it up, and they insert it in the groove of self molecules. These are called MHC or HLA, human leukocyte antigen. That's important, and I'll show you why in a second. But it presents it on the surface of the cell to the T cells. This CD4 cell, remember, is a cell that's infected by HIV, but there's a subset, there's a clone of CD4s that come in and through their specialized receptor see a piece of HIV in the groove of this MHC protein. Then they're the maternal cells of the immune system. They're the helper cells. They secrete some pro-inflammatory cytokines that make themselves better at what they're doing. They make this cell better at what it does. Then they interact, and this is where it gets important, they interact with CD8 positive T cells. These are the assassins of the immune system. Their job is to go out and kick ass, to find an infected cell, to go up to it, interact with it, and deliver a death signal to it and kill that cell. That's the most important thing that these cells do. But because it's not good for the host to have killer cells floating nilly-willy around the body, because then you're going to have autoimmune disease, it's highly regulated. And the first step in that regulation is that it's got to recognize the virus in the groove of a self molecule, of a self MHC, of a self HLA, and seeing just one small piece of the virus. It binds like a lock and key kind of fit, and then it gets activated. It says, okay, I finally found my target, and it kills it. It does a few other things that I'm going to show you in a second. But the bottom line is this axis is the immune system now fighting back and trying to get hold of the virus. The problem is it's happening too little and too late in most people, not in people like Sandy. And I'm going to show you that in the next couple of slides. This is what happens. So we're getting at a higher and higher level. If I was taking blood out of somebody who had just gotten infected with HIV, The virus shoots very high. It's crazy. It goes, you have, that's on that axis over there, 1 million to 10 million copies within a week to 2 weeks of getting infected per milliliter of plasma. That's a lot of virus. There's a lot of virus in the body. It ramps up, and then there's this point where it comes back down. There's an inflection point where it comes down. This drop in the viral load, the peak viremia, corresponds with an increase in CD8s, those assassins I just told you about. The interesting thing is, in everybody, the CD8s come up and bring the virus down. But depending on how strong the CD8 response is, the depth that this plunges is determined by the strength and the proximity and the kinetics of that. In most people, though, it hovers around 50 to 100,000 copies in the blood. So it's lower than the peak, but it's not low. Not like Sandy. Sandy and her group, it comes down to undetectable levels. It's still there. The virus is not eradicated. It hides out in sanctuary sites. But it comes down to incredibly low levels. In most people, though, during this 10 years or so in the middle, there's this phase of clinical latency. The virus is there. It's still, you still have it in you. It's revving up the immune system. It's replicating to high degrees, but the immune system can keep pace with it. And then what happens is over time, this erodes. The CD4s creep down, and as they get low, opportunistic diseases kick in, and those are aptly named. So these are opportunists. These are things that everybody is exposed to. Your body, you get infected with them, Epstein-Barr virus, cytomegalovirus, herpes, some of the herpes viruses, pathogens that are fungi, yeast that get into the body, don't cause much problem. But when you become immunocompromised, those things wreak havoc and people die from those. And they're typically things that, as I said, that are kept at bay. So this is what happens inexorably if somebody doesn't start antiretroviral therapy. In other people where the viral load is low, that's probably less likely to be an issue, but not everybody is free from that. There are some people that still progress. So this was a slide we made a long time ago and we never used it in anything, but I think it's an interesting way to illustrate that if you think of the time to disease progression or people getting HIV and ultimately developing AIDS, the median time is 10 years. So this is why it's a lentivirus. It takes a really long time for it to really screw up the host immune system. There are unfortunate people who 48 months into infection succumb to an AIDS-defining illness. So these are rapid progressors. There's a genetic predisposition for that. At the other end of this are slow progressors. These are people, if you map out their CD4s, instead of doing this like the average person, they kind of do it at a much different slope. But eventually they have to start therapy. And then there are the people like Sandy that we put here. And we actually think that they might even be on a different distribution because they might never progress. So the median duration of infection in my cohort is 29 years. We have two people that were confirmed to be infected since 1978, since before many of you were born here, and we still cannot find virus in them. So unbelievable outcomes. As the AIDS pandemic continues to expand, the need for an effective vaccine has never been greater. Durable control of HIV replication does occur spontaneously in these rare individuals, as Sandy pointed out, very rare, less than 0.5% of the infected population. They go by different names. We still call them what we've been calling them for a long time, which is long-term non-progressors. And we believe that understanding the mechanisms of control in people like Sandy is the key to cracking this nut, figuring out how to control the virus, how to cure people from HIV. So to do that, we wrote this protocol. I'm the principal investigator of it. And to be enrolled in this study, you have to meet very stringent criteria. You have to be truly positive by standard antibody tests. So it's not a false result. These patients test positive antibodies that are reactive with HIV in their body. Healthy with stable clinical courses, no history of opportunistic diseases, non-declining CD4 counts, viral loads below 50, and not receiving antiretroviral therapy. So really hard. It's hard to meet all those criteria. Sandy meets them, and the 135 people we have in our cohort meet them. So I like to throw this up. When I meet the patients and I see them, I try to see them every visit. Sandy can attest to that. One of the first questions I always say is, why do you think you've done so well? Why do you think you're still here? Because there's a lot of emotion that comes with this. People have survivor guilt. They've buried a lot of people they love. They've seen their friends die. And they come in and they say, I want to contribute something. So I say, well, why do you think that you've done so well? These are quotes from patients. Sandy's not one of these quotes. But one person, I exercise religiously and eat well. I did smoke 2 packs a day. I drink 2 liters of Diet Coke and never exercise. I have no clue. This was real. I was feeling depressed and drank rat poison when I was in my 20s. I'm convinced that's why my immune system's different. This woman still believes that to this day. One of my favorites, I drink only the most expensive red wines from France. It's because of my family support, my positive frame of mind. And then a lot of people say this, which I think Sandy echoed when she talked. I'm just lucky. God's given me a gift. It's got nothing to do with what I do. I'm just blessed. So it's probably wired into your genes. That's a predisposing factor. People also say, researchers usually say, well, why do the patients, you're not helping them. NIH people go to NIH because they want to get a treatment they can't get anywhere else. So what do you do for them? Well, we give you a little bit of money to defray incidental expenses and to pay for your inconvenience, but none of them care about that. They come here because they get insight into their disease course, they're contributing to an important cause, they're paying tribute to lost loved ones, and most importantly, many of them have mentioned to me, they feel that they're serving as beacons of hope for their children and future generations. So these are an altruistic group. People are not coming to NIH to make money or to be famous. They're coming because they want to contribute, because they want this to go away. So we're all on the same page about that. So, and this is easy to read slide. The point of this is the first 64 people that we included. And this is just to pay homage and tribute to all of their contributions. So these are men and women, as you can see, who have been infected, as I said, for a median duration of 29 years. They come from various racial and ethnic backgrounds. They've acquired HIV by different modes of acquisition. They have normal CD4 counts and low viral loads, as I mentioned. And in an extraordinary small study that we did, where we used an experimental assay that is not available commercially, that measures the amount of virus down to 1 copy in the peripheral blood, we found that a third of them have less than 1 copy. And the median viral load in these patients was 2 copies. Sandy was one of the ones that was less than 1 copy. Median viral load is 2, which is exactly what it was in age-matched controls, patients who were receiving antiretroviral therapy. So extraordinary, extraordinary control over the virus without taking medications. One of the first clues we had that there was a difference was we checked for HLAs. And I told you, these are the self-proteins that bind to pieces of the virus and present to the T-cell receptors of CD8s. This is the most polymorphic locus in the gene pool. Right now, the probability that any of us have the same HLA type is very, very low. So we haplotyped all these patients, and imagine our surprise when we found that one of them, HLA B5701, was present in the first 11 of 13 that we tested, and it continues to be present in 70% of them. So it's neither necessary nor sufficient. It's not in 100% of them, and there are people with B57 who have progressed, but it's an important clue. Sandy carries B57. She's B57 positive. So this was a clue that CD8s, because of that cartoon I showed you at the beginning, and for time's sake I didn't put in other slides, these present to the CD8s. So my job when I first joined the lab was to try to understand how CD8s could be different. So, and this was the point I was going to make when I was when I was struggling without slides. So of the factors that you could think about, virus factors, could the virus be weak? There have been reports where inactivating mutations in key regulatory genes have been documented in some controllers. There have been reports of the virus being pummeled by the immune system in one person and being transmitted to a recipient, and then that recipient has an okay course and the virus isn't as high. And there's been some data supporting maybe the virus was just weak. But I can tell you, in the majority of people we study, this doesn't matter. So Sandy is not as well as she is, doing as well as she's doing because she is infected with a weak virus. Her virus is fully virulent. We can tease the virus out and use it to infect other cells in the lab. It's her immune system that's what's different. So the host genetics, I always get asked by scientists, oh, it must be this mutation in CCR5. CCR5, I told you, is the co-receptor that HIV needs to get into the cell. And some people have a mutation, this delta 32 mutation in it. And if they do, they have a lower expression of the co-receptor. And there were some early reports back in the day saying, if you have that, you're more likely to control more slowly, to progress more slowly. That's been debunked. We found that that's not the case, that most people, Sandy does not have this mutation. We can come back to what that means because there's been a lot of press, because people that are homozygous, that have 2 copies of this, that have no co-receptor, are resistant to infection. That was discovered in Rwanda by studying commercial sex workers. That is the source of the Berlin patient, who is the only case of sterilizing cure. He was given a bone marrow transplant with somebody who had 2 copies of this defective co-receptor, and any HIV left in his body had nothing to infect, and it went away. And he's the only case of cure. But the other host genetic factors that are important are protective HLA alleles. And I already mentioned these are the self-proteins that decorate the surface of every nucleated cell in your body, which is basically everything but red blood cells. And in disease-free situations, it presents healthy peptides, and the immune system swims by and says, oh, you're cool. There's nothing wrong with you. No cancer here. no virus here, they keep going. But if that cell gets infected by a virus, and a piece of virus gets inserted there, or that cell gets transformed into a malignancy, and a malignant peptide shows up, the immune system knows it's different and pays attention and interacts with that cell. So this is the HLA associations I alluded to before are suggestive or supportive that the immune system is important. And as I mentioned, CD8 positive T cells preferentially target these HLA peptide ligands that sit on the surface of the cell. So really the pole is in the middle. What matters is how the CD8s interact with the infected cells. So I'm going to skip all this and I'm going to go to my this is my first animation slide ever about 15 years ago. It's horrible, so I'm embarrassed. But so this is basically a lot of data that I'm going to get right to this. We had evidence that the CD8s that in patients like Sandy kill much more efficiently than the CD8s of everybody else. So my students joke with me that I spent the rest of my youth trying to figure out how to measure killing of CD8s in the lab. And what we found, this is from one patient on the top who is a controller. This is from one patient on the bottom who is not a controller. We take out their CD4s and we infect them with a form of HIV that has green fluorescent protein in it. So it shows up in its image this is confocal microscopy. So this is like a microscopic image. Everything that looks like the snow globe, all these white dots, these are infected CD4s from that person that I've been able in the lab to have them express lots of HIV. Then I put in, on top of it, CD8s from that same person, an image every couple of minutes. This is a really cool technique. And then we can do really quantitative measurements. And what we see is by 15 minutes, all you see is schmutz in the non-progressor. All those little dots, I can gate on them and use software and identify regions of interest that are real cells. They get wiped out really fast. That's not the case for a progressor. Their CD8s see the virus, but they're not good at killing. There's some defect in their ability to kill. So we've been able to do this over and over. We have 5 or 6 different assays that show that. Also, I thought you might enjoy seeing a movie that we use the same confocal microscope to do. These are cells from a non-progressor. This is actually Lorine. These are Lorine cells. So everything in blue are CD8 positive T cells that have been stimulated with HIV for 6 days to get them revved up so they could see the virus again. Then we count them and we label them with a tracking dye that makes them look blue. And then we add fresh infected cells, and these are cells infected with an RFP or a red fluorescent protein virus, so it looks red on the imager. And we put them there and we let them co-incubate, and these cells go crazy. The blue cells go nuts. They see the red cell, and over time you see this one at the top interacting with it. It basically bites a hole in it and the cell dies, and it dies like that. When you put progressor cells there, they interact, they pull around. Sometimes the cell dies. They usually detach. And the blue cell moves on to some other red cell, interacts with it. Why is it that it sees it, but it can't kill it? It's responding to it. It recognizes the HIV. It's not because the HIV is so mutated. It will make some antiviral proteins in response to it, but it just won't kill the cell. And so this is what we have many projects ongoing in the lab. I'm not trying to sort that out. So if I've bored you, if I've said too much stuff, this is the money shot. This is the one that is really the final thing that I want to tell you. So in this slide, this is basically on the top, this is a scenario that I think is the cascade of events that you need if you're going to control HIV. Everything I'm going to say about the top row applies to Sandy cells. So when we take CD8s and we mix them with various antigen-presenting cells, cells that have virus on them and can present virus to them, and we let them do their thing in a dish, in a well, in a flask, all sorts of ways that we do it to trick the cells into thinking they're in the body. We give them just enough CO2. We do it at 37 degrees. It's really unbelievable that you can mimic those conditions. We stimulate them for 30 minutes, even up to 12 hours. The cell from a person whose immune system doesn't control the virus and the cell from a person whose immune system does look exactly the same. In the first several hours, they get revved up, they get activated, they express some antiviral proteins that are kind of wimpy proteins that do a little bit to keep the virus at bay. They don't do a lot. Really what happens that's significant is what happens over the next couple of days. In a long-term non-progressor, I'm sorry I'm going late, when a long-term non-progressor cell comes into contact with the virus, the cells go through a proliferative cycle. They get revved up. My brother loves it when I say it's like Agent Smith in the Matrix movies, that he's a really effective assassin, and then he replicates himself in wherever the second or third one it was, replicates himself, and all of those can really, they're basically having a big army full of heavy artillery that can really do do what they need to do, and pick off all the cells in the vicinity. So when these cells come into contact with a bunch of virus in a lymph node or in the gut or in other sites where the virus is replicating, they're able to bring the virus down to really low levels. And then they kind of look around and wait for other infected cells. And when they don't see it, they shrink back into a few sentinels that percolate through the body looking for another upheaval. Very important. And this is really done very efficiently. One of the questions you should be asking is, well, why is there any virus at all left? The virus hides in certain sanctuaries, and it's integrated into some resting cells. And we're working on trying to figure out how to make your CD8 so good that they're able to go in cells that are hiding the virus, they can even eliminate those cells as well. So the goal would be to cure somebody. SANDY is considered a functional cure. We want to do sterilizing cure and purge all of the HIV from her body. In a progressor, it's a very different scenario. So this is a person, most people who have HIV, what happens over 3 to 6 days is very different. These CD8s see the virus. They're alive. They're interacting with it. They're trying to figure out how to tackle it. They secrete some antiviral proteins. They divide a little bit. These yellow dots are supposed to be the killing granules. They have killing proteins, and when they one of my goofy cartoons was them liberating the granules, and the granules migrate across, put a pore, through a pore in this cell, get in and deliver a death signal to the brain of the cell, and the cell dies. So these divide a little bit. They look like they should be good killers. They don't kill. They just kill just a little bit. And so we've found several different ways in the lab to show this, that they'll kill, but most of them interact with the virus. The virus keeps amplifying and amplifying. They keep it at bay. They do enough to hold it to about 50,000 copies, but over time this exhausts this immune response, and the virus wins if you don't start therapy. So I think that's really the last slide I was going to show. So I'm going to skip all these because I said it a lot and I said it fast, but the bottom line is the last bullet really is the unifying feature of what our collaboration and the collaboration with all of the people in your group that we talk about is understanding how Sandy's immune system is different so that we can figure out how to apply this to people who aren't as fortunate, so we can figure out how to make an immune-based therapy or a vaccine that's therapeutic or prophylactic, and or help other people with other immune-related diseases, and there's a lot of them. So we have kind of a big mission that we're trying to grapple with. And so my acknowledgement slide, and the most important group, I have a great boss, Mark Connors, who we have a great working relationship for a long time, but the people I always give the most credit to are the patients who make this work possible, which are the patients. So, thank you. Any questions in the last minute? Sandy. Yes.
So you said that you never really got sick before, and so what made you get tested since you never really?
My husband died.
Oh, okay.
You know, he died of AIDS. So actually it was a situation where I went to visit him in the hospital and he was being rushed down the hallway. And I was like, what's happening? Where are we going? He had been in the hospital for a couple of months. And we didn't know what was wrong with him. So that's what life's so awful. Everything was totally wrong, and they didn't know why. He was in the hospital for 3 months. And I went to visit him, and they were rushing him down the hallway. And that's when I got the news. They said, have you been tested for AIDS? I'm like, what? What are you doing now? And my husband had AIDS. And I was totally blown away. It gives me goosebumps even, but I remember the feeling in the moment. So I went and got tested. And when I got tested, to my surprise, I didn't think, no, not me. Sure enough. So get tested. Know the answer. Oh, you want me to?
Yeah.
Oh, what the heck? Excuse me. Okay.
How old was your daughter when you found out that you were HIV positive?
That was 1989, 10 years. She was 10 years old. Well, actually, at the end of 10, she was going almost 11.
Do you ever think that the blood samples of people who are elite resistors and stuff like that would be used like kind of like how HeLa cells have like?
Yes! Oh, I'm sorry. I'm so blessed I mentioned Henrietta Lacks and the HeLa cells. Most of you did not see TV and watch Henrietta Lacks on HBO with Oprah. Did anybody see that? 1, 2, big all right, great. I couldn't wait for it to come on. You guys don't watch TV. I was like, studying and stuff. But I feel very much compromised because I was ignorant for my status and I would have been able to help sooner. That's my main problem. Like, why keep me in the dark? What are you doing with my blood, large draws of blood, and you're not telling me that I'm able to help or doing something that's nothing wrong with me? Just, excuse the expression, sucking me dry for what? for nothing. I'd rather be doing something to give back, if I have value. I didn't know I had value. I don't know. I don't know.
A question about the long-term non-progressors. Could it be possible that their CD4s are simply easier to kill, the infected CD4s are easier to kill? Is that a possibility?
That's one of the things that we, and as controls in our papers, because we're always looking, is it on the CD8 end, is it on the T cell end? In that cartoon I went through quickly, one of the things that we ruled out a while ago is that the cells are not resistant to infection, so they're very permissive and susceptible to infection. Then the other question was, do they just die more readily, which is what you're asking. And we have no reason to think that's true with the control experiments that we did, where we did crossover experiments and we looked at different things. Is there a mechanism of dying? It's an apoptotic death, so it's a programmed cell death that's induced by the same factors. When you use other stimuli to kill them, there's not any increase in that. When we culture them for prolonged periods of time, the viability and the recovery we get at the end of time is exactly the same. It depends on the viral load, though, going in. So when we correct for the amount of virus, because they have more virus in their cells than non-progressors do, because the virus has been hit so hard for so long, it's a little shy, whereas in progressors, they have more virus around because there's just more that's being replicated. When we correct for that, there's no difference in this than the ability of the CD4s to die. So we don't think that's the case.
But have you tried taking progressor CD8s to see if they can kill the non-progressor CD4s more easily? Or?
So we did that. That was actually in our paper in 2008. So the problem with that, as you know, is that when you take heterologous cells from different people and they see HLA, they know that's why tissue gets rejected, right? Because you're seeing foreign HLA. So there's going to be alloreactivity anyway. But we controlled for that, and we were able to, above background, measure any extra killing in infected fours. And we were trying to determine where the pole was. Is it on the target end or is it on the effector end? And exactly what you said, when we put progressor cells on long-term non-progressor targets, they didn't kill that great. When we put non-progressor CD8s on progressor targets, they killed exactly the same as they kill their own targets. So it really doesn't support that.
We understand that her immune system is extraordinary. She doesn't get sick. She doesn't get anything that a normal person can get. Can you briefly explain why other diseases, such as cancer or other high profile, diseases, won't affect someone like her?
So that's not entirely true. So there are certain things, so the two historical facts I always get from it's really incredible because these patients come from all over, they have all sorts of different backgrounds, and the two things they always tell me are, I never get a cold. When I'm in the office and something goes through the office and everybody's out sick, I'm the only one who never gets sick. And the other thing they always tell me is I have really rapid wound healing. If I get cut, if I'm in an accident, everybody marvels at how rapidly I heal. And there are kids that have the same genes that have, usually with HLA-B57, will report the same thing. But you're not completely impervious to everything because in our cohort, a subset of people are co-infected with hepatitis C. There's no difference in hepatitis C outcome in non-progressors. They don't control hepatitis C more readily than the typical person with hepatitis C. So this effect is virus specific. It's HIV specific and maybe in some cold viruses. We don't think there's some other viruses that we don't think it has an effect on hepatitis B. The cost of having it is that you are also susceptible to some immune reactive diseases. So B57 and a gene that sits right next to it that's in linkage disequilibrium with it is C6. And those two together are enriched in cohorts of patients with psoriatic arthritis. So you can have an autoimmune inflammatory arthropathy related to that genetic background, which isn't good. That could be very debilitating. So it's kind of like this spectrum, this is how I think about it. There's a spectrum of immune responsiveness. And the good news about having a really robust immune system is you can control some really tough viruses, but the cost is that you can also suffer from immunologically related diseases. So it's not perfect. In our cohort of non-progressors, there are people on the West Coast who really believe that they are all eventually going to progress. They also think that the little bit of immune activation, inflammation that they have because of their immune system having to interact with HIV once in a while is more than a healthy person who doesn't have HIV, and it's going to predispose them to early heart disease, early liver disease, early kidney disease. There's some, in my opinion, weak data that supports that that's the case. But it's really, when we have looked at we have the largest international cohort, and we've looked at their health over time, and we've compared them with other patient groups. We're not seeing increased cancer, we're not seeing early heart disease, we're not seeing, so I do think that they're relatively protected, but they're not resistant to everything. There's not. We've had some skin cancers. We've had a few people with peripheral vascular disease, a few people with stroke. So that's why I tell Sandy, one of the most important things I tell them is, you are going to probably die with HIV, not from HIV, but you have to control everything else. Get your diabetes under control, get your blood pressure under control, get your cholesterol down, do all those health maintenance things that you should be doing, and your HIV is probably going to not be a problem. In our cohort, I should tell you too, 5% of the people that we follow have progressed, meaning their CD4s were flat and then they started coming down very gradually. And we're trying to understand why, but it's only a handful of people. So we haven't been able to find anything that seems to account for why they've progressed.
I noticed that in your initial sample of non-progressors, the 64, that there weren't any Asian or Indian ethnicities. Is there a reason, or is that just that sample?
So that's a tough question because it's still a fairly small cohort, even though it's one of the largest, as I said. And as we've grown it more, we have more Hispanics than we did initially. We have no Asians still in it. There is some evidence in Thailand, because the Thai Red Cross is a really impressive setup, that maybe the HLA associations are different there. So we know B57 is associated with control in sub-Saharan Africa. We know, especially in South Africa, where there's been a lot of work on it. There's evidence in Europe that it's associated with control, and definitely in North America and South America, it's associated with control. But in Asia, it doesn't seem to be the case. And it seems that there's other HLAs that are responsible for that. But the studies have been very small, and I don't think anybody feels confident drawing any conclusions yet about it. But that's a good pickup. We have wondered, we should collaborate with other people with other patient compositions to see, but everybody kind of has the same composition we do, so we're not sure if it's cause and effect, what's cause and effect.
So is there a difference in the proteins that poke the holes in the virus between the long-term non-progressors and the ones that progress?
Outstanding question.
Allows them to destroy them faster versus...
Outstanding question. We've sequenced the perforin, and we've looked at granzyme B and by a number of different assays to see if there's something different. There's so many things to probe. We're just now, we've been doing this for a long time, and we've been working with a lot of postbacks, but we're just now looking at things like, if the cell, is it that the perforin can't get out? That it degranulates to put it in the immunologic synapse where it's going to float over and affect the target? Is that the cells, as my one student said, are they constipated? Can they not just get the perforin out to do its thing? That doesn't seem to be the case. Is the granzyme B that's getting exocytosed, is it dysfunctional? We came up with this assay that I was really impressed with. My boss thought of this way that we could do it, to look at the activity of it once it got out of the cell. Looks exactly the same. It has the same activity between a progressor and a non-progressor. So it's really not that. It's this whole series of events that I showed in that last cartoon of, if they don't load, they have to divide to load. There's something about the body's evolved a way to prevent you from becoming a good killer without making you go through cell cycle and turn on some genes that you need to turn on, and do it gradually. So it's almost like you have to pay your dues to become an effective killer. And it's a slower process. So with people that don't control, the defect starts early. They don't divide as well, they don't load their proteins as well, then they don't kill. We don't think it's a problem with the granules getting out. We don't think there's a dysfunction in the proteins themselves. It's just the way they're regulated. We are looking at other things to get at that. So it's a complicated, you're asking a very complicated question really, because there's a lot of nuance to it. And we're looking. We haven't closed the door on most things. But that one thing, the fact that perforin, the sequence of it looks identical, there's still, you could argue, maybe the way it's processed, maybe post-translational modification, there could be something that's different that way. We're still struggling with that, but we don't think there is based on preliminary data.
I have a question for Allison. What sort of advice would you give to somebody worried about getting tested?
Worried about getting tested? Don't worry about it, just do it. Just do it. Don't worry about it, just handle it. Just go take care of it. Just go in. You know, it's quick, fast, they'll give you the results now. I think like in 10, 15 minutes you can get the results back. Just do it. You want to call me? Take my number. I'll go with you. Anybody want to go? I'll go with you and make a ride.
I can understand how this research could lead to treatments, but how could a vaccine come out of understanding how non-progressives work?
I love that question. Did I plant that? Did I ask you to ask me? Right now, the pendulum swing, like in a lot of things, swings back and forth. 10 years ago, we would go to our big HIV immunity conferences, and it would all be T cells, and nobody would talk about antibodies. Now some neutralizing antibodies have been identified in some people. So these are antibodies that bind to the envelope that I showed you, to GP120, and they block its ability to infect the CD4. And everybody said, oh, this is really important, because if we can make a really good vaccine that does that, it's basically would be like a flu vaccine. It would induce an antibody to bind to it and prevent it from getting in. The problem is you're going to have resistant mutants. You're not going to cover all strains and all clades. And the risk and the downside is much more significant. If you get HIV, but if you get you get flu, you're sick for a few days and then you clear it. So we know that it's not just antibodies, but that's not where the pendulum is. Everybody's talking about antibodies and cure and control, and that's getting a lot of hype. Louis Picker, who is one of my heroes, who works at Oregon Health Sciences, has a rhesus macaque model, and they've vaccinated these monkeys with a CMV, which is what humans get. 90% of the people in the room are CMV positive. It's a human herpes virus. So they've vaccinated monkeys with macaque CMV, carrying pieces of SIV, which is the simian homologue of HIV. And they've shown that when they come in and challenge these monkeys by intrarectal challenges or by even putting it in the blood, 50% of the monkeys are not only controlling it, they're being cured. They're eliminating it. And they're showing that it's only being mediated by CD8 positive T cells. There's no induction of antibody responses. So the skeptics and the critics could say, well, that's only 50%. But that's the best signal that I've ever seen. Of all the studies that have been done, that's the best thing. So that to me is a really important piece of information, that CD8s, like the CD8s in Sandy, the unifying feature is they see the virus, they recognize many different pieces of it, and they interact with it in a very important way, and they're very functional, and they're responsible for eliminating it. There's some differences with Louis Picker's model, and I don't want to get into the details, but I'm really making the loose connection that it's the same cell type that's controlling a viral infection that we thought was resistant to control. So those are the types of clues that we've gotten over time, which is what we can ignore. We know it's not the innate immune system. We know that there are elements of the humoral immune system that are unimportant, but we've nailed down the players and the prospects. And I think that's what, because when I looked at my notebook when I first joined the lab, the possibility was a huge list. And over time, we've really narrowed it to a few things, thanks to studying cells from Sandy and her colleagues.
What steps you guys are going to find the cells where the HIV are dormant in and hide, isolating those and just get rid of them?
Yeah, so this whole thing about purging the virus, shock and kill is one of the catchwords, where you stimulate the immune system, you try to smoke all the virus out, and then you find some way to eradicate it. Nothing's been successful thus far. Drug companies are very interested in this. There's a lot of pharmaceuticals that are being devised to not activate the immune system, but be just potent enough to tease out the virus at a decent enough level that it can be seen, and then kicking in the CD8s to then kill those cells. I haven't seen anything yet that looks really convincing, but that's the goal in the field is if we could figure that out, then we can cure. That's how you eliminate or eradicate HIV, is if you can get rid of the reservoir. We have a collaborator at Penn that we've been working with for a very long time now, she has looked at the cells in our patients, and contrary to what the Parisians found when they looked at their controllers, in our controllers, she found that their reservoirs were smaller than people on therapy, which was shocking. But which shows over time that even though it might be changing, that is, you're laying down these things that are getting stored in the brain of the cell, right, in the nucleus of the cell, that even though it's changing, something is actively seeing them and periodically removing some of them. And we think it's the CD8s. So I think what we're doing will inform strategies to purge the viral reservoir, to answer your question. We're not there yet. I'm not trying to be grandiose. We have a long way to go. But that's kind of part of the goal of what we're doing.